YouTubers, welcome to this video tutorial. It will cover various aspects of vegetative propagation. It will be dealt in several parts. Today we will discuss the part 1. Part 1 will cover stem cuttings, types of stem cuttings, collection, procedure of making cuttings, handling of cuttings, their treatment with rooting hormones. So, first of all, let us understand what we mean by the vegetative propagation. Vegetative propagation in plants is a technique of plant multiplication through vegetative parts. It is an asexual method of plant reproduction. A new plant is produced by artificial vegetative reproduction techniques, which are referred as macropropagation and micropropagation. Macropropagation involves cuttings, layering, grafting, and suckering, and use of vegetative parts like stem, roots, and leaves. Specialized reproductive structures or vegetative propagules include rhizome, tubers, suckers, and micropropagation involves tissue culturing. So simply, vegetative propagation in plants is an asexual method of plant multiplication, which can be done through cuttings, stem cuttings, root cuttings, or leaves, or through its vegetative propagules. First of all, what we mean by the cuttings? A plant section originating from stem, leaf, or roots. So cuttings can be from stem, it can be taken from leaf, or it can be from roots, which are capable of developing into a new plant. So plants developed through cuttings are called as clones. It is adapted for multiplication of horticultural and ornamental plants, the most and efficient means of multiplication. Is employed by gardeners to grow new plants, both deciduous and evergreen plant species. Is most popular among plant propagators, farmers, and is considered as the cheapest and the most convenient method of multiplication. So the cuttings can involve the stem, leaf, and roots. And the plant which is formed through it, new plant which is formed through it, is named as clones. What are the parts used? As we have already mentioned, stems can be used for vegetative propagation, roots can be used for vegetative propagation, leaves, modified stems, tubers, corms, rhizomes, runners, bulbs, and these are all parts of plants which can be used for vegetative propagation. This video will mostly cover the stem cuttings, the propagation, vegetative propagation through stem cuttings. What is a stem cutting? Stem cuttings are the outgrowing stems, or mature sprouts or shoot cuttings, a portion of the stem, cut and planted in the soil. Stem cuttings develops adventitious roots and then they grow into a new plant. That means you are taking a stem cutting which can be taken from the outgrowing stem, a part of a stem, a portion of a stem, and then which is then planted in the soil. It forms an adventitious roots and grows into a new plant. What are the types of stem cuttings? According to the nature of the wood, stem cuttings are classified as herbaceous, non-woody, softwood cuttings, semi-hardwood cuttings, hardwood cuttings, and these reflect the growth stage and the type of the stock plant. So we will discuss them one by one. Herbaceous, softwood, semi-hardwood, and hardwood cuttings. Herbaceous non-woody cuttings. Shoot cuttings or tip cuttings taken from young soft tips of stems during spring season. They are easy to root in short span of time, produce high percentage of adventitious roots, and stem measuring 3 to 5 inch long is cut from the parent plant. Leaves on the lower 1/3 to 1/2 of the stem are removed. Few leaves are retained at the upper end as a conservative measure to conserve the water losses there. Examples where the herbaceous non-woody stem cuttings can be taken: carnations, geranium, triticale, chrysanthemum, and dahlia. These are the examples where from the herbaceous non-woody stem cuttings can be taken. Here are some of the typical herbaceous stem cuttings which are here. One is the geranium or pelargonium species. This is gladioli, and which is chrysanthemum, and there is purple heart, and then is also the triticale, which are both types of the triticale here. These are some of the softwood herbaceous stem cuttings taken for propagation. Softwood cuttings. Here are some of the softwood cuttings, as this is nerium. The cutting is taken of this size, 3 to 5 inches, and you are removing the basal leaves to conserve the water, and this is the cutting which can be planted or which can be treated. Semi-hardwood, semi-ripe or SR cuttings. They are prepared from partially mature wood of the current season's growth. Cutting collection season usually is from mid-July to early fall. Size of the cutting 3 to 6 inches. Leaves retained at the upper end to lower the water loss and closer spacing in the rooting bed. So it has two purposes. Why you reduce the leaves? Why you keep the upper leaves only? Because you are planting these cuttings together. When there will be less lower leaves, so there can be closer spacing in rooting bed. They can be planted in a very closer space. And there will be also the less water loss because you are removing the leaves which otherwise will face the transpiration losses. Large size leaves are generally trimmed, rooted under cool and moist conditions. They are maintained by intermittent mist sprays. Since they are very tender and they need and moisture, they should be sprayed with intermittent mist sprays. Examples include broad-leaved evergreen ornamental shrubs, euonymus, camellia, azalea, citrus, and some trees also, citrus and olive, are also semi-hardwood cuttings. So semi-hardwood or semi-ripe cuttings are taken in the month of mid-July to early fall, and then they are trimmed. Their 50% of the leaves are removed. The top leaves are kept. If they are larger, they are trimmed. Here is how you take the evergreen or the semi-hardwood shoot cuttings. So you are removing the shoot tip, upper shoot tip is removed, and then lower leaves are removed, and you are also taking sometimes if it is a side branch, you are taking the heeled cutting, and it can be a main branch. This is the side branch. So here you are removing the lower leaves, and also if it is the terminal portion, soft tip is trimmed also. So this remains as a cutting. The upper few leaves are retained, whereas the lower leaves are removed. So cut made nearer the lowest bud. So this is how you can make the evergreen or the semi-hardwood shoot cuttings. Now the fourth type of cutting is the hardwood. Hardwood cuttings include, they are dormant, fully mature stems, deciduous shrubs, and trees taken at the end of a growing season. winter or early spring. So they can be collected at winter, or that is the end of the after the autumn, or they can be taken at the early spring. The wood is firm and does not bend easily. Deciduous shrubs. These are some of the shrubs and the plants which will have an hardwood cuttings. Poplar and the willow are popularly propagated by hardwood cuttings. So poplar as well as the willow, they have the hardwood cuttings, and these privet, fig, grape, pomegranate, quince, these also have the hardwood cuttings. So hardwood stem cuttings having 4 to 30 inches in length is collected, should be taken from a healthy stock plant growing in full sunlight. Cutting should be taken from the tips of 1 year old. So remember, you have to take the cuttings from the current year, not from the old stem, current year shoot. The cutting should have sufficient supply of stored food material. Usually central and basal parts of the stem should be preferred. So we are preferring for cuttings from the hardwood, the central and the basal part, and the aerial part is discarded. How you make the cuttings? You have to take a pruning scissor and you take the stem of the current year growth. This is the current year growth. You are cutting it here, stem cut with a sketcher, and then you make a cutting here having several nodes. Now we have an uppercut and the lower cut. Uppercut can be, for the distinction, you can have an uppercut as an oblique cut just above the bud. So it should be few centimeters above the bud, the uppercut. While as the lower cut should be nearer to the bud, it can be straight cut below the bud. So the two oblique as well as the straight cut makes it distinct to see the polarity of the stem cutting. Because when it is deciduous, it is very difficult to locate the basal portion or the terminal end, because that time the buds are not there visible, and at that time only the cut, the oblique cut, will be treated as the terminal part, and the straight cut will be taken as the basal part. So this is how you are taking and trimming of the stem cuttings for propagation purposes. Evergreen plants, stems should be defoliated, left only with few leaves at the top. Avoid cutting with flower buds. Remove any flowers or floral buds on the cutting. 2 nodes are included in the cuttings. Upper cut should be few inches above the first bud, as I have mentioned in the diagram, and the lower cut nearer to the axillary bud. Lower cut nearer the node increases the chances of successful adventitious rooting. In large scale operations, bundle of cutting materials are cut to desired lengths by mechanical cutters. So when you have to do it on the larger scale, that time you have to have an mechanical cutters where they can be cut in bundles. A standard length for a stem cutting is about 5 to 6 inches. Medium sized stem cuttings goes 20 centimeters long. And there are now 3 types of the hardwood cuttings, or the cuttings which can be made: the straight, heel, and mallet type. They are preferred, and we'll see what is the difference between the 3 types of these cuttings. Straight cutting is the most commonly used stem cutting. To differentiate between top and the base of the cutting, make one cut at the slant rather than at right angles. Mallet and heel cuttings are used for plants more difficult to root. Heel cutting retains a small section of older wood at the base. Mallet or hammer cuttings are entire section of the older stem wood, or a piece of the twig is cut together with the stem. Additional piece of the twig helps to develop root system. So we have 3 types: straight cut, heel cutting, and mallet or hammer cutting. They can be shown this way. This is a straight one, where you have kept the lower cut and the upper cut. They are straight. Then you have a heel where you have taken the piece of the older stem with it. At the base of it, it has the base of an older stem also with it. This is the current year's stem. While as in mallet, you are taking a branch, a portion of the branch of the older stem. It looks like a mallet because this helps us in the formation of the adventitious roots here. So these are the types of the hardwood cuttings: straight, heel, mallet, and then it is from these basal areas where you can see later on adventitious root formation is taking place. So what is the procedure for making stem cuttings? Cutting should be cut from a healthy stock, because healthy stock in a nursery is maintained. Stock plant should be always healthy, that can give you a healthy cutting material, and it should be taken from a healthy stock at 45 degrees angle. You have to use a pair of sterilized sharp knife for secateurs, and select 1-year-old shoot branch or a sub branch, strong, healthy and disease free, with no discoloration, toxicities or deficiencies. So you have to be very choosy when you select the propagating material. You have to inspect the stock plant. You have to see that it is disease free. It does not have any toxicities or deficiencies which can be evident on the plant. So for stock plant which are difficult to root, there are certain practices like etiolation or girdling are applied. So the two types of the plants are there. One is easy to root and another is difficult to root. But difficult to root definitely roots, but with certain more practices. Etiolation and girdling are the two practices which will be discussed in the next slide. Nutritional status of the plant is altered before collection of the stem cuttings. What is etiolation? As you know, the etiolation, as a science student, etiolation means the growth and development of plant in absence of light. You'll see the plant turns yellow. It's characterized by long, weak stems and small yellowish leaves. Where you are growing the plants away from the light, etiolation effect remarkably increasing adventitious root formation in stem cuttings. That means there is a relationship between the light and something going on with the stem cutting, that is with its hormone concentration. Light has a negative effect on root initiation. It interferes with the root promoting action of endogenous auxins. So that means when it has the cutting has internal endogenous auxin, that time when the light falls on it, it has an effect on it. It interferes with the root promoting action of endogenous auxin. That's why when it is kept in darkness, there are more chances of the development of the adventitious roots, because auxin is not disturbed, is not affected in absence of the light, but in presence of the light there can be a deterioration in the levels of the auxins. This is how it is done. It's practiced. Etiolation is you just take in small this house and cover it. Blackout tents can be used to cover stock plants. You have a stock plant where from you have to take a cutting, you cover it, and to generate etiolated stems, and then this is how the etiolated stems looks, because they will be yellow in color, they will be elongated, and then you take these etiolated and then you plant and cut them and plant them in a soil or treat them with hormones, and you see the difference. This is a non-etiolated, so root formation is less. This is an etiolated where you see the plenty of the roots are available here. So that means etiolation is taken to the advantage of rooting. It's done by putting the stock plants under cover in tents, so that blackout tents, so that they undergo etiolation process. Girdling is a technique. It refers to the constricting or ringing of a stem by removing bark along with the phloem below the aerial foliage. You know the stem. Stem has internally a phloem and a xylem. Whatever comes from the leaves, that is moving from that has a polarity moving from the upper to the lower, from source to the sink. And what is done here, a ringing or a constriction is done on the stem, and which blocks or binding the base of shoots with the wire for several weeks before taking the cutting. Above the girdle, what you experience the stem, there is a swelling due to accumulation of carbohydrates, hormones, and other root promoting factors, resulting in an increase in root insertion. When you are cutting, you are ringing a plant. Since from source to sink there is a downflow and it is restricting the movement there, so there will be accumulation. When you are taking that stem that has a more hormones, more carbohydrates, and the chances of root insertion is more. So cutting taken from the juvenile parts of the plant can better produce roots compared to the older parts. Ideally, there should be 1 or 2 nodes at the top of the stem cutting. So girdling is done this way. You are making a girdle here. You are making a girdle here by removing the portion of it, and it is fine girdling. This is wide girdling. This is 50 days after girdling, where you find after 50 days of girdling, there you will see the swelling of the upper portion, because carbohydrates, hormones have accumulated there. So this area is taken and it will have the chances of more root formation will be there. This is how girdle is made here, and this is some girdling controls here. Because of the girdling control cuttings with callus formation, this is whenever you are not girdling it, this is a rotten control cuttings. You have taken two types of the cutting, one which is control, which has not been subjected to the girdle, and the second one which has been taken from the girdle areas where ringing has been done, and you see the difference. They are rotten here. They have produced girdle cuttings with callus formation, and after the callus formation there is a rootings of these cuttings. That means girdling has a positive impact on the root formation in the stem cuttings. If a stem cutting is taken from this girdled area, it will have more rooting adventitious root formations, while as if you take it from the ungirdled, they are mostly rotten, and that means girdling and the etiolation are the two techniques which are helping in the formation of adventitious roots in the stem cuttings. The question is how we will collect and how you will handle the stem cutting material. You have to take the stem cuttings from healthy stock plant. This is the first and the foremost rule. And you have to take it from the juvenile parts. And you have to take the shoots from the bottom, the lower shoots, and collect it early in the morning or late afternoon. And you use a sterilized pruning scissor or a scratcher, and take material which should be kept moist, cool, and turgid. So either wrapped in a moist burlap or kept in large unsealed polythene bags. And during hot sunny days, cutting should be frequently sprayed with mist. So whenever you are going for a large scale or a small scale, you have to collect a cutting material. It has to be collected early in the morning or late afternoon, and then you have to take the precautionary measures to keep it moist, cool, and turgid, and you can also spray it with the mist. Herbaceous plant cuttings should be treated stuck soon after being taken. Because they are fragile, the cut end should be immediately submerged in water. To assure good hydration, cutting should be packed in plastic bags. To conserve water loss, you have to trim, as we have already mentioned, each leaf to 1/4 of the original size. And sever each cutting cleanly. Make a clean cut. Thumb rule for stem cutting is that you should have a cut just below a bud at the base and just above a bud at the top. Why we are keeping a bud below cut just below a bud at the base? Because it is the bud where from the adventitious roots will be developed. Why we are keeping the above cut where the bud is at the top? just above a bud. The top cut is above the bud, because there are every chances that that area, the upper area above the bud, can get dehydrated and can be lost, can be degenerated, and but the bud will be preserved there because it is away from the top cut. So this is the thumb rule for stem cutting. It's advisable to take basal lateral shoots. Lateral shoots have low nitrogen, high carbohydrate balance, and which is favorable for good rooting. And then you take the terminal shoots. Terminal shoots have the high nitrogen and low carbohydrate ratio. Several cuttings of uniform length are tied together in a bundle. This is in the larger scale, and bundle is buried horizontally or vertically with basal ends. How we will differentiate the basal end? Because you have made a straight cut, and at the top you have made the oblique cut. 6 to 8 inches of sandy soil or sawdust, especially in colder regions. So when you are taking the bundles of these cuttings, you are putting the basal end in, they are buried in a sandy soil or in a sawdust, and this procedure promotes root insertion at the base and retards bud development at the top. So to facilitate, there should be the callus development at the cut end. Basal ends of cuttings are buried under moist sand for 1 week. So that means when you are taking a cutting, there should be a callus development. A callus arises from the cells of a cambium, that the basal end of the cambium, basal end of the cut. So when you are putting them and you bury them under a sandy soil, moist sandy soil for a 1 week, there is a chances of the callus development. Or you can put them in other media as clay pellets, rock wool, coir, vermiculite, perlite, and soil. We have already discussed these soilless mixtures. The callus is a soft tissue formed over a cut plant surface, arises from the cells of the cambium. Some of the callus cells on stem cutting gives rise to roots. So callus formation leads to the root formation, especially the adventitious root formation. So this is how, if you are taking the rose cutting and you are putting them lower basal end in a sand, after few weeks you will find this white coloration is appearing there and swelling is appearing there. This is a callus formation. This callus formation this is the callus formation, and it is the area where from the roots will originate. That roots will be originated from the callus cells, because these are the cells of the cambium, and this is how a callus will lead into the root formations in the cuttings. So that means first and foremost is the callus formation, which is done by putting the cuttings in a sandy, moist sandy soil kept horizontally, and it develops callus formation there, and then it leads to the formation of adventitious root formations. In refrigerated rooms, cuttings can be safely stored under cool, moist conditions at a temperature of about 4.5 degrees until spring. In early spring, cuttings are dug up, inserted about half their length vertically 3 to 4 inches in trenches. Prior to the hormone treatment, cuttings should be treated with mild doses of fungicides, benomyl 0.5 grams per liter against fungal infection. So that means you have taken a cutting, either you have kept them in a refrigerated room or you have directly then taken them in a soil, but you also treat them with the fungicides so that they are disease free, and then there is a treatment of the cuttings. How you will treat the cuttings now? There is a treatment that can start. The basal portion of the cuttings are wounded by removing bark from each side. So this is before the callus formation. Before you are putting them, you can also treat the cuttings, which will facilitate the root formations. You are wounding by removing the bark from each side. Wounding made with sharp knife or a razor blade. A vertical cut measuring 1 to 2 inches is made with a sharp knife, penetrating through the bark at the basal end of the stem cutting. Commercially, a uniform and rapid wounding is done with the help of device having 4 sharp prongs. So wounding promotes root production. It is effective in juniper, rhododendron, maple, magnolia, holly, and it has been seen by Willis in 1962. So treatment of the cuttings is also one of the treatments is also called wounding. So you are taking the basal cut with the help of a sharp razor. You remove a thin layer of a wood on this side and the second side also. So layer of wood removed to aid rooting. This is called as the wounding. Making wounding cuts in the base of the stem, cutting to stimulate rooting. This is also helpful in rooting. Now, after that, you are using rooting hormones because there are some species which are hard to root or they will root less. To promote rooting, a farmer or a gardener or a propagator uses the rooting hormones. How to use the rooting hormones? You have to take rooting hormones. They can be in a liquid form or they can be in a metallic form, and then they are to be applied at the basal ends of the shoot cuttings, which can be difficult to root stem cuttings, and they will be treated with root promoting substances. The auxin, they are of the auxin type. Basal ends are dipped in commercial talc preparations also, or soaked for 24 hours in dilute solutions of synthetic rooting hormones like indole acetic acid, indole butyric acid, naphthalene acetic acid, and 2,4-D. These are some of the synthetic hormones. Though there are also natural preparations which are also promoting the rooting, but here we are focusing on the types which are having which are auxin type and are synthetic in nature, which are proving good for promotion of the adventitious roots in the stem cuttings. This is the chemical structure of the indole 3 acetic acid, indole 3 butyric acid, naphthalene acetic acid, naphthoxyacetic acid, and 2,4-D. These are some of the synthetic hormones which are used for rooting the stem cuttings or promoting the adventitious roots in stem cuttings. Hormone treatment stimulate first root initiation and also promotes uniformity of rooting. It also increases the percentage of rooting. Before using rooting hormones on a desired plant material, a simple test named as tomato leaf cutting sensitive test is performed, because you have to see what will be the concentration which will be effective, what type of hormones are more effective. For that there is a primitive test that's called the tomato leaf cutting sensitive test. This test is tomato cuttings are sensitive to growth regulators. A plant propagator can use tomato leaf or a stem cuttings to check the efficacy of a plant preparations for its rooting promoting properties. Simple and sensitive test for effectiveness of rooting hormones. What you do, you take a stem cutting of a tomato plant, and then you put it in a hormone concentration for soaked for 24 hours, and then you see within this liquid rooting hormone there will be the formation of the rooting, and this is how the rooting will be visible on these stem cuttings of the tomato. So this is how to grow tomato. They can be grown through the cuttings, but here it is used for making the test for root promotion. So treated and untreated tomato cuttings are inserted in a moist sand, kept in polythene covered containers. After 2 weeks, tomato cuttings, both treated and untreated, observed for the percentage of rooting production. So, to determine the best material and optimum concentration for rooting in a particular species under a given set of conditions, empirical trials are necessarily carried out. So, you are using the tomato cuttings to understand what will be your requirements, what will be the percentage of root production, and that will help you to use the hormone for your desired plant species. Other, other talic preparations. Talic are the powdered forms. Talic preparations are available with proper protocol, and they also show the list of the plants which are responding to certain treatment, to talic treatments which are available in open market. Before dipping the stem cuttings, a fresh cut at the base is mandatory for sufficient adherence of the powder. So you are using an exogenous this powder form of the hormone so that it should adhere to the stem cutting. You are making a fresh cut, or you can take the bundle of cuttings, can be simultaneously dipped in the talic powder, and you have to take a proper care. All cuttings in the bundle should receive a uniform amount of powder preparations. To avoid contamination in talic powder, take a limited quantity of a powder in a container, or else you can dip it in talic and then take it in a water solution also. There are two ways which we'll be discussing in the next slide. So you have a talic preparations which are available, which are used for promoting the rootings in stem cuttings. Some of this is a Ceradex, which can be used for this hardwood cuttings, and this is Ceradex 1. It can be used for soft cuttings, and same way there is a 2 which will be used for the other types of the cuttings. So you have the powdered form of the hormones available, which are specific to types of the stem cuttings which promotes rooting in them. Suppose here you see a nerium which is a cutting is taken, and then you are stripping the leaves, keeping only the aerial leaves of it, and this is the overall cutting. You have a Ceradex. You can put the Ceradex in a water and dissolve it, and then you can take the cutting which is already having in which all the leaves have been removed, lower leaves have been removed or defoliated, and then you are putting them into this medium, which is basically a powdered form, but it is made as a dilute concentrations of it, and then you are putting them. After few weeks you will see the rooting formations there, and then they can be taken and planted in a pot. The soil mixtures are there, and then you are ultimately taking this the rooted one and then putting them into a pot and then watering it thoroughly so that the newly rooted stem cutting is established into a new plant. So here is the use of talic powders, or you can use the talic powder also. You take a stem cutting, dip it in a water, and then put it in a Ceradex mixture or a talic mixture, so that the talic mixture gets adhered to this area, and then take it back to the water where it is and keep it there for 24 hours, or depends upon the species, and then plant it and then see the root formations. So these preparations are used here to improve the rootings. Treatment with liquid formulations. As you have done with the talic, there are also the treatments with the liquid formulations. Plant propagator can prepare a wide range of concentrations. When you have to prepare, you take 1 liter of 100 parts per million solution, and there you have to take 100 milligrams of a pure grade rooting hormone. It's dissolved in 10 milliliter of ethyl or methyl alcohol and diluted with water up to 1 liter. This is how you make a concentration of 100 parts per million. And NaA being directly insoluble in water, few drops of ammonium hydroxide are added before adding water. So to prepare 100 milliliter of a 4000 parts per million concentrated solution of a root promoting substance, a 400 mg of pure chemical is dissolved in 100 milliliter of 50% ethyl or methyl alcohol. This is how you make the preparations or the concentrations of certain liquid formulations, which are basically the rooting hormones, which can be IAA, which can be NAA. So basal part of a stem cutting treated with liquid formulations, the stem cuttings can be either soaked in dilute solutions or dipped in concentrated solutions of root promoting hormones. Depending upon the species, you know whether it is to be dipped or it's to be soaked. Dipped can be in a concentrated solution or soaked will be in a dilute solution. In both these methods, bundles of cuttings can be easily treated at a time, allowing all cuttings to absorb or uptake uniform quantity of hormones, promoting uniformity in the results. Because you are putting a bundle together and they will absorb uniformly and there will be the uniform results. The same solution can be repeatedly reused if it is tightly sealed in a bottle to avoid evaporation of the alcohol, so which would otherwise affect its concentration. So this is how you are making the liquid formulations and how you are putting them, either you are dipping them or you are soaking them. Dilute solutions of indole acetic acid should be immediately used owing to their rapid deterioration. So if you are making an IAA concentrations, they are to be used immediately, otherwise they will be deteriorated. IAA in a sterile solution remain active for several months. IAA under unsterilized solution is readily subjected to bacterial destruction. Dilute solutions of IAA should be used immediately owing to their rapid deterioration. Uncontaminated solutions of NAA 2,4-D can maintain their strength for as long as a year. So that means different hormones have their different storage history. NAA as well as 2,4-D can remain for a long time, while as IAA under unsterilized medium, it will last for a very little time and can be subjected to rapid deterioration. So as I said, there is a soaking method and the dip method. In soaking method, basal end of cuttings are soaked in a dilute solution of rooting hormones and maintained at about 20 degrees for 24 hours away from the direct sunlight. IAA is sensitive to light. IB is more stable than IAA, while NA and 2,4-D are entirely light stable. And IBA, NAA, 2,4-D are resistant to bacterial decomposition and light destruction. And you maintain effectiveness over a long period of time than IAA. Easy to root species can be soaked in dilute solutions of 20 parts per million. You see the concentration matters here. It's of lower concentration because they are easy to root species. Difficult to root cuttings can be soaked in solution having 200 parts per million. That means they are soaked. Here they can be kept in dilute solutions when they are easy to root species. Difficult to root needs more concentration. So this is how you are putting the cuttings for a long time under soaked conditions, because they are to be kept there for a long time so that they imbibe, they absorb more of the hormones. This is how soaking is done with the cuttings, and the different concentrations of hormones are applied here, which are already trialed because knowing what concentration will give the best rooting results. The dip method, which is the last method here, as I said, involves quick dipping of a basal cutting in a concentrated solution, varying from 500 to 1000 parts per million. So it's for a short duration, only for about 5 seconds. So here what you do, you take step 1, this is how you mix it first. In second, you put it and dip it, and then after that you take this cutting and then plant it, and then it will develop the rooting there. So here it's concentrated solution. Dip-in has a concentrated solution, while as the soak-in, where you soak the cuttings, has a low concentration. So that is what we have seen so far. We have seen in this video the vegetative propagation, various aspects, that what are the types of the stem cuttings on the basis of their nature, and then how they will be collected, how they will be treated with different hormones, whether in talic form or in certain liquid formulations, and also how they will be treated like wounding, etiolation, girdling techniques. We have seen in this part 1 up to the treatment and handling of the cuttings. Hope this video will help you to understand the things, and in the next coming video, that will be the part 2. It will be a series of the propagation that will cover the rooting medium, that will cover the other aspects of the propagation, and will cover the stem leaf cuttings, root cuttings, and the other aspects of vegetative propagation. Hope you have understood, and this video will help you to understand the propagation techniques. In case you have liked this video, please subscribe the channel. Till the next video, goodbye, be happy. Thank you very much.